Hello and welcome to today's webinar. On behalf of Drug Target Review and Beacon Discovery, I'd like to thank you all for attending. I'm your moderator, Victoria Rees, Deputy Editor of Drug Target Review. Today's speaker will be Dr. Carlton Sage, Vice President, Computational Sciences at Beacon Discovery. Following his presentation, we'll be moving on to a live question and answer session where you can pose questions you might have for today's speaker. You're able to submit questions at any point during the webinar, and you can do this using the questions panel in the menu on the right. So without hesitation, may I pass over to today's keynote speaker, Dr. Carlton Sage.
Thank you, Victoria. So we're going to talk about drug discovery in the early phases of drug discovery and trying to differentiate between high-throughput screening or virtual screens. We're going to focus on two examples. The first is an example with a well-studied receptor, Serotonin 2B receptor for systemic sclerosis. With this receptor, there's going to be a lot of information in the background, lots of known chemical structures, crystal structures available to help us find new ligands for alternative indications for this receptor. The second is a receptor in which we know almost nothing, basically some expression data around an orphan receptor called GPCR 174. It looks like it has potential in immuno-oncology, but we don't have any structural or chemical information available. So let's start with the 5-HT2B receptor. First, I want to step back and give you an overview of Beacon Discovery and the team's track record. Beacon Discovery as a group has been working on GPCRs for about the last 20 years. In this time, we've put more than 30 compounds at the clinic doorstep. 20 of these compounds have actually made it into the clinic. One of them has already been approved, lorcaserin, for chronic weight management, and that's a 5-HT2C agonist ligand. Importantly, Beacon has also worked on orphan receptors, and the orphan receptors that we've put into the clinic are, first off, we discovered the natural receptor for the niacin ligand. And secondly, we've put compounds for GPR119 and GPR52 into the clinic with collaborators from Johnson & Johnson and Boehringer Ingelheim. So we believe that GPCRs represent a significant untapped opportunity still. Overall, there are 750 GPCRs that are expressed in the human genome. About 135 of these receptors are targets for approved drugs in the United States or the European Union. And it's a huge market. About 700 approved drugs target GPCRs, and that represents about 35% of total approved drugs. Some blockbusters that hit GPCRs are Tagamet, Plavix, and Abilify. There are still untapped opportunities within this class of receptors. The 350 traditional GPCRs include 120 receptors with no known ligands. These are called orphan GPCRs. And only a handful of these have been targeted to date. There's also an additional 350 olfactory GPCRs that have yet to be targeted. And there's one last category that we're going to kind of explore a little bit more today, that GPCRs which already have ligands that have liabilities, which we can address by further understanding how the receptors signal and design ligands that only modulate the pathways that we're interested in modulating for the disease of interest. So, like I said before, Beacon works on all aspects of GPCR drug discovery. We have very flexible collaboration terms. We can work either as a drug discovery incubator, or we can help you understand the literature and other aspects of your targets of interest. We also have an internal portfolio of drug discovery programs that are available for collaboration and out-licensing, and we can provide to your company additional bandwidth and breadth to your partner's GPCR drug discovery efforts. And the area that our talk is going to focus on today is really project enablement. We're going to be trying to find new molecules that help modulate the activity of our receptors so that we can develop tools to prove whether or not our hypothesis of disease treatment is valid, and furthermore, develop these ligands into molecules that we can then do lead optimization and hopefully become clinical candidates. So like I said at the beginning, we've got two projects. We're going to start with the 5-HT2B receptor, and we're going to try to find antagonists for this receptor to treat systemic sclerosis. Serotonin receptors is a well-studied class of receptors, and they have a wealth of structural and chemical data available. What we're going to leverage here are huge compound libraries, a big compute engine, and our existing robust assays. So of course, when you're trying to have a drug discovery project, you want to focus on helping the patients. And here we decide to go after the serotonin 2B receptor for the treatment of systemic sclerosis. A little background in systemic sclerosis. It's an autoimmune disease that's involving microvascular damage, dysregulation of innate and adaptive immunity, and generalized fibrosis in multiple organs. And when patients go see the doctor, the first thing is usually noticed is skin fibrosis. That's what leads them to further understanding of what problem they're having, but it's the pathological changes in the lungs and other major organs that determine the clinical outcome. This disease affects about 2.5 million people worldwide, and there's no approved medications to address it. So why go after the serotonin 2B receptor? Well, we know that serotonin is involved in the fibrotic pathways. We know that 5-HT2B signaling is one of the components of this pathway, especially the inflammation pathways like TGF-beta, and it represents a potential important therapeutic target. There's also additional literature that suggests that serotonin activity at the 2B receptor is a mediator of pro-fibrotic activity. So serotonin receptors are a big class of drug targets. It's a group of 14 receptors which bind serotonin, which is also known as 5-HT or 5-hydroxytryptamine. Nine of the receptors that interact with serotonin are G protein coupled receptors. We're going to focus on the class 2 receptors, which is a family of 3 receptors. For this class of receptors, there's rich structural information. We've got 15 crystal structures of the receptors in all states of activity, either antagonist state where they're kind of going back and forth between active and inactive, but they aren't allowed to signal, inverse agonist state where the receptor is locked in an inactive conformation, and then the state of the receptor as an agonist with an agonist bound, which is where it forms its functional activity. So our approaches for new ligand discovery are going to be using ligand-based or structure-based approaches to discover new ligands. Now, we're blessed with all this structural information. However, there are challenges. Because this is a well-studied class of receptors, finding novel chemical matter with lead-like properties is going to be hard. In addition, these receptors are highly homologous. So getting selectivity of action against the 5-HT2B receptor versus the 2A and 2C receptor is going to be important as well. So one of the first things that we have to do when we're evaluating a virtual screening approach is to look at the crystal structures. And we all understand that when we look at pictures of crystal structures in journals, they're beautiful, exquisite models of the receptors. And yes, they are beautiful, exquisite models, but the important part is that they are all models. They're all just mathematical representations of the data. So we have to dive a little bit deeper into our understanding of what these models are actually telling us when we're picking a a target in which to try to dock molecules. So in this slide, we're going to compare two crystal structures, 6DRZ, which is a serotonin 2B receptor with methyl surgide bound. It's a relatively high resolution structure. It's got good crystallographic parameters. But when we take a look at the electron density, and that's really a visual representation of how well the model recreates the experimental data, we notice that for the ligand, there are parts of the ligand, as you can see, there's a hydroxyl pointing out into the screen that is not well covered by electron density. So we don't know whether it's an unoccupied space, it's a space of the ligand that's moving around a lot, so we really don't know where that hydroxyl is pointing. So we have questions about the structural usability. Compare that to the 6DSO structure, the one on the bottom, which is a 2B receptor with LY266097 bound. Again, pretty approximately the similar crystallographic parameters. But when we further explore how this ligand is bound in the binding pocket, the ligand density is almost complete, meaning that we have a more confidence that the conformation and position of that ligand is more certain. So for this project, we're going to decide to move forward with a 6DSO structure and not use the 6DRZ structure. Another important aspect of virtual screening and docking is when you can do it, to have the ability to do a test of how well your selection method, be it docking or 3D shape and conformation with a known ligand, how well that can remove or identify ligands that you know to be active from a set of property matched decoys. So we're going to compare our methodologies using a property match set of decoys that have been built based on several thousand decoys that were created from known ligands to serotonin receptors. This hasn't been just for 2B antagonists, but serotonin receptor ligands in general. And we're going to evaluate it using two methodologies. First, it's called ROCKS. This is a software tool from OpenEye Scientific that looks at shape and chemical features of a ligand and when it compares them. And we're going to compare the ability of these ligands using this tool to pull out active compounds from our decoy set. And for the second experiment, we're going to use docking using the 6DSO structure. And we're going to ask questions like, should we use the basic nitrogen constraint? So almost every serotonin ligand has a positively charged nitrogen that interacts with an aspartate in the binding pocket. We're going to move away from that for two reasons. Number one, we're looking for antagonists, and number two, we want to have a wider space of ligands to consider because we know that the patent literature is relatively crowded. And there's two ways, well, there's one way to look at how we're going to do this. We're going to use what's called a ROC curve. And this ROC curve shown in the upper right-hand part of the slide is a methodology that was invented in World War II by radar engineers when they were trying to look for enemy planes versus flocks of birds. And so they invented this receiver operating characteristic curve comparison. And a perfect receiver operating characteristic would be essentially would look like an L that's turned sideways, where it goes just straight up and then horizontally across at 1. We don't get that kind of performance with virtual screening tools. And we're going to compare these two ligands in terms of how well they remove decoys and select active compounds from the decoy set. As you can see for ligand 1 on the left, we have a pretty good ROC curve. It's not perfect by any means, but we are getting a performance above random. This black line running bisecting the rectangle is random performance in terms of selection. Now, ligand 2, on the other hand, doesn't perform well in terms of selection. And we think there are some probably pretty reasonable rationales for why. It's a small molecule, it's highly charged, and there's probably not going to be there's going to be a lot of ways for it to be similar to other compounds. Now for docking, we're again trying to select active compounds from a decoy set, and we wanted to consider a couple other things. First, we wanted to ask how well does it perform where we put all the constraints of the ligand in, like the shape of the pocket being a little bit more constrained, the positive charge being required, and you can see that that curve looks pretty good. Now, it doesn't get too much worse when you take out the ligand, and that's for the full-size pocket. The additional consideration we had to make was that these computations are huge, and they scale pretty much linearly with the volume of the binding pocket. So if you can reduce the binding pocket by reducing the binding pocket volume, you can save compute time, which means saving money. So the lower set has both the ligand-based bias pocket and the empty pocket, but the volume has been reduced by about 4-fold. And we get relatively similar, although you could argue that it's a little bit worse performance, but we thought that the cost savings was worth the relative drop in performance. So we decided to go with this reduced volume binding pocket. So the last thing before we press go on this experiment is to talk about the ligands that we're going to be searching through. And I just wanted to step back and give a little bit of historical perspective on what we've been able to do from a computational perspective compared to what ligands we had available at the time. And the two have kind of been running in parallel. So when I started doing these kind of approaches back in the early 2000s, the number of compounds that you could buy was about 1 million. And the compute power that we had available was individual machines. We didn't have even clusters at that time. So we had to essentially just set up runs on our own personal workstations. These were mostly SGI visualization stations as well, and do the docking experiments, and they would take days to go through 1 million compounds. If you move ahead a decade, 2010, now there are about 25 million commercially compounds available. It was kind of a pain because you had to go to all the different vendors and put that collection together internally. But once you did it, you had a little bit more compute power. There were small clusters available. So we could then still do our docking experiment in days using our internal cluster against these 25 million compounds. The interesting thing was that the docking experiment and the small molecule comparison experiment, this 3D shape and chemical feature comparisons, took about the same time in those days as well. So again, days to go through 25 million compounds. Jump forward another decade. Now the commercially available compounds are approaching 5 billion. And the compute power, I have it in quotes, is unlimited. I mean, it's not really unlimited. You're not going to be able to take all the compounds that we think we could make. The drug-like chemical space is about 10 to the 60. We're not there yet in terms of compute power. However, for the scale of computations we need to do, it is unlimited, because for the 3D shape and chemical feature comparisons, the compute times are hours. And for the docking experiment, the compute times are days. So we've got about 2,500-fold increase in the chemical space we can evaluate computationally in the last 20 years. It's pretty amazing, and in large part, it's been the work of companies like OpenEye Scientific to leverage this compute power on cloud platforms like AWS. So here's an overview of exactly how the computations work. So for our ligand-based virtual screening, we start with a 2D structure. It gets conformationally generated into a 3D structure, and you map the shape, and then you map the chemical features on the outside. And then you use these shape and chemical features as a probe to search. At this point, this was done about a year and a half ago, 1.5 billion compounds from the enamine reel collection. And this took about an hour. So our approach with the results was to take the top 500 compounds, cluster them by scaffold. Sorry, we took the top 500 clusters and then picked the scaffolds that had more than 20 members in them to then select for compounds to buy and test. The second experiment is the docking experiment, where we took our model from the 6DSO crystal structure and docked 1.5 billion compounds from the enamine reel collection into that model of the binding site. This took 3 days. And at the time, we're not 100% sure, but we think it was probably the largest virtual screen ever done in the world. We pulled 10,000 virtual screening hits, and we selected scaffolds from this set with greater than 20 members, and we ended up with 31 scaffolds. And it's a little bit, when we ran this experiment, it was pretty fun because it's a little bit small on the slide, but at one point we had 35,000 computers running in parallel to get this job done. And just that, thinking about what that room must have sounded like to get this job done was pretty fun. So how do we go about, from these thousands of compounds that we've virtually screened, what to purchase and evaluate? So we wanted to think about it in terms of how we can enable our medicinal chemists if some of these compounds turn out to be active and look like they're going to have the kind of properties we want to develop. So we basically looked at the molecules in terms of the core that they represented, thinking about how the substitutions on the outside of the core, R1 and R2 here, have been changed. And then for each core that looked like it had good chemistry on the outside, we evaluated the properties that were represented in the compounds that we had. And if those properties looked relatively drug-like, then we selected that core to purchase a subset of to evaluate experimentally. We also discarded scaffolds that had a lot of IP literature around them. And then we identified our purchase sets and we ordered them. So a summary of the overall experiment. For docking, the input was this 442 cubic angstrom non-ligand biased pocket. We ordered 71 compounds. We received 56 of them. And the difference there is, one of the great things about this enamine reel collection is they haven't made all these compounds yet, but they know they think that they can make them based on the chemistry they have. So that's why there's a small reduction in the number of compounds that are available. We tested these against all 3 of the related serotonin 2 receptors. We got 22 hits. A hit was defined as a compound that was 5 micromolar or better. It was a 40%, roughly a 40% hit rate. And the best hit was 22 nanomolar. For the search based on shape and chemical features, we had 2 literature compounds as inputs. We ordered 39 compounds and received 30. Again, tested them against the serotonin 2 receptors in-house. We got 11 hits, about a 37% hit rate, and the best hit was 2 nanomolar. And the overall results of this whole project was that we identified 2 novel chemical series, one from the shape and chemical feature approach, one from the docking approach. And the team, including the medicinal chemists, saw that these ligands were worthy of further exploration, and we continue to work on them as we move forward. Okay, so that was an example of a project where we had a lot of structural information to start with to help us find new chemical matter. Now we're going to talk about GPR 174. So that's an orphan receptor. It has potential immuno-oncology implications, but we don't have any structural chemical information available. So a little background. So GPR 174 is a class 1 orphan receptor. It's expressed on a variety of immune cells. It has high expression on natural killer and Treg subsets of these immune cells. And its nearest neighbor is the P2Y10 receptor, roughly 50% identity. It's reported to respond to lysophosphatidyl-serine, but this has been difficult to replicate in the lab. We know it's a GS-coupled receptor that has high basal activity and constitutive activity, and it's likely relevant in immuno-oncology. So there's some things about the receptor that lead us towards this hypothesis. First is that elevating cyclic AMP in immune cells is typically strongly anti-inflammatory, and this can also lead to immune suppression in the context of tumor biology. So Beacon collected a treasure trove of expression data around both primary immune cells and immune cell lines. And the reason we highlight this is because if you compare the expression of GPR 174 in all the immune immortalized cell lines, you can see that it's very low compared to the primary cells isolated from tissues. And this is comparing FKPM data from RNA-seq studies. When we develop our assays to do high throughput screens, we have to consider having the expression of our cell lines match that of the primary cells. And you have to be really careful because immortalized cells, as we know, often have different expression than what you see in the normal tissues. So for any high throughput screen, assay development is really key. And robust assays, as you might expect, are going to be the key to getting good answers from your high throughput screen. We express our receptors in HEC293 cells and CHO cells. We label the receptors with an N-terminal tag to follow cell surface expression, and this is used in most of our orphan GPCR cell lines. And all the HTS campaigns we perform at Beacon are done using clonal cell lines. And at the bottom of this line, we just have a demonstration of what it looks like once we clone our receptors into the HEC293, get a big shift in expression on the cell surface. And you also are given, at the end of each experiment, because we do clonal selection for development of the cell lines we use to test, you get to choose from expression levels of the receptor you're interested in. And this can be really important. Some receptors that are highly expressed and highly constitutively active, you need to look for lower expression. And of course, those that aren't highly constitutively active, you want to have higher expression so that you can see a response from those receptors. So as far as chemistry, it's really important that the libraries that you screen are diverse and contain compounds that you can get a hold of afterwards. Beacon, now part of the orphans family, has 600,000 compounds available to screen. and they explore a wide range of chemical space. The libraries are pretty heavily GPCR focused, probably a third GPCR focus, small focus on kinases, and the rest are drug-like diversity. We also have a roughly 1,400 compound library of fragments that we can screen. Now for this project, we screened our drug-like diversity set. The receptor was screened in inverse agonist mode. We looked at the blockade of cyclic AMP accumulation following addition of a PDE inhibitor. The screening project as a whole took roughly 2.5 weeks, and that included the pilot screen plus 3 full days of HTS. That was going 60,000 compounds a day. The resulting data was very robust, a really good separation between the positive control and the negative control, a good C, and a good standard deviation on our response data. We selected about 4,500 compounds for follow-up reconfirmation. For those 4,500 compounds, we were reconfirmed in triplicate at 10, 2, and 400 nanomolar. And we also had a control assay to look for compounds that were non-selective or hitting something else in the HEC293 cells. At the end of the day, we selected a set of 75 compounds that have confirmed activity and complete selectivity of the control assay for further follow-up. So following up on our hits, we really believe that a reconfirmed active with evidence of selectivity has to be confirmed using fresh powder, and that either comes from resynthesis or repurchase. These authenticated powders are then used for 10-point dose response curves versus additional control assays. And once we have compounds that are active against, in this case, our target receptor GPR 174 and the similarly signaling decoy receptor, we declare those compounds as hits. We also have developed assays for the rodent orthologs along the way, and these are really important because as you head down the drug discovery process, you're going to need an in vivo species, and you want to know early whether or not there's going to be shifts in potency between your species that you're comparing. We profile each compound in orthogonal assay platforms, for example, arrest and recruitment. And we use this to increase the confidence in our hit set. For GPR 174, we currently have dozens of compounds that are confirmed, and we're starting to work on a medicinal chemistry plan. Okay, so what happens now that you have hits? And this is where the real fun begins. The team gets together and develops an initial target product profile. It depends on the route of administration, target organ, and other properties. The team evaluates properties of the existing hits and identifies liabilities, for example, intellectual property, ADME properties, in vivo model activity, and such. Then the medicinal chemistry team and the computational team begin to evaluate ways to expand the hits to find the most efficient path to an in vivo proof of concept tool, either by doing synthetic expansion around the hits, and usually combining that with visual screening of available chemical libraries. So now we're pretty much in the same spot with each of these two projects. First, one with hits we got from virtual screening. Second, GPR 174, where we got hits from a real experimental high-throughput screen. And now we're moving forward with developing these targets into drug discovery programs to help patients. So one last slide about Beacon Discovery's capabilities. So as you can see, we've got lots of experience working on all aspects of GPCR drug discovery. We've got extensive experience in interrogating novel GPCR therapeutic opportunities, and we've worked all across the board from target validation and hit to candidate development. We have an extensively characterized high-throughput screening compound collection of more than 500,000 compounds. And from this set, we can eliminate unproductive compounds based on our history of screening these compounds and immediately focused on the interesting chemistry. As you can see, we use state-of-the-art computational tools for virtual screening and hit expansion. Our database that we have internally has powerful tools for evaluating our hits and validating them. And we have access to a wide variety of receptors where we have profiling assays already set up so that we can rapidly determine selectivity for new receptors and prove on target activity. And we have significant med chem and computational chemistry experience and a large in vivo pharmacology group that has extensive small animal pharmacology experience. And then one last slide to really say that this presentation couldn't have been possible without this group of people in front of our buildings in San Diego, California. And of course, this photo was taken before the COVID-19 outbreak. We've worked together as a team. Pretty much everybody in that picture has been together working on GPCRs for the last 20 years. We put 30 compounds at clinic doorstep, 20 have been into the clinic. We've evaluated more than 50 GPCRs as drug discovery targets. We've got 100 patents, around 75 papers published on GPCRs, and we continue to push the boundaries of GPCR drug discovery. With that, I'll say thank you very much.
Thank you, Carlton, for your excellent presentation. We'll now move on to our question and answer session where Carlton will be answering all of your questions. Don't forget, you can still submit any questions you might have using the questions panel in the menu. So let's take a look at the first question. So, Carlton, were you able to identify agonists at GPR 174 at the single concentration stage when adding the high throughput screening library? And there's another comment that presumably the CAMP gas tone in the assay was not outside the linear range and therefore it was possible.
Oh, hey there. Thanks for the question. So yes, we have been able to get hits for GPR 174. Right now we are in the hit follow-up stage. And in this hit follow-up stage, what we do is first, after we do the single concentration test, we'll do the dose response. Well, actually, we'll do 3 more single point concentrations. And if we get activity there, then we kind of pause and we acquire the compounds and to get physically new samples. So we either make them or purchase new samples. At this stage with 174, we have acquired probably about 100, 150 compounds for GPR 174, and we have the powders in-house. And we are currently working on dose responses for those now.
Great, and yeah, just a second part to that question. Could you confirm if these were or will be followed up? For example, any cluster analysis done on potential agonists versus inverse agonists, and also including the option to use a GPR 174 agonist as a challenge compound against inverse agonists identified to deconvolute binding site?
Yeah, absolutely. So those are all great questions, and we typically run all of those kind of experiments internally to look at both inverse agonist, and inverse agonist versus antagonist approaches once we identify agonists. I do believe we have agonists and inverse agonists identified, so we're going to continue working on these as we go.
Fantastic, thank you. So next question from our audience now. Could you confirm that the other GAS specificity assay had the CAMP tone to understand confirm hits were not inverse agonists at another constitutive active receptor?
Another great question. Yeah, we always look at similar signaling receptors when trying to identify and prove that our hits are actually doing what we think they do.
Great. And are there any plans to progress confirmed GPR 174 hits into a phenotypic / native assay at all? And if so, what cell line is this?
Yeah, absolutely. We're going to put them into primary cells and eventually into animal models. Yeah.
Great. So next question now, how many compounds do you need to screen in a high throughput screening campaign to get any meaningful results?
So that's a kind of a tough one to answer. It really depends on how hard the receptor you're going after is. Typically, we tend to approach it from focusing on how good the assay is to begin with, and secondly, trying to be as diverse in our compound collection. Right now, we've kind of settled on about 500,000 compounds as the sort of good size of a collection to screen, to not necessarily guarantee you're going to get hits, but to give you a really high likelihood of getting hits.
Great. So how do you identify real hits from off-target hits or even promiscuous compounds?
That's a fantastic question. We use several approaches to do that. First, when we're acquiring our libraries, we try to focus on compounds that don't have any properties that would lead them to be promiscuous. These kind of the pains properties that you read about in the literature. The second approach is we always have an orthogonal screen that we run alongside that we know is a robust one that we've run many times in the past to look for compounds that are lighting up both assays. They're obviously going to be promiscuous. And then the third way is just the robust dose response curves for each of the compounds.
Great. So yeah, please do keep your questions coming in. We've had some really excellent contributions. So the next one now, Carlton, for you is, can homology models be used for virtual screening?
Yeah, absolutely. For GPCRs, as you probably know, the number of crystal structures is growing, and it's fantastic work in the literature. But the number of crystal structures is probably less than 20% of the total GPCRs that are out there. So we've been experimenting with using homology models, leveraging the already determined GPCR structures, and have had some pretty good results using homology models, as long as the template for your homology model is close enough to make it a reasonable model to use in a virtual screen.
And next question now, given the wealth of high throughput screen data tested against a number of targets, if a new target is tested with the high throughput screen collection, are Beacon able to provide a data warehouse for specificity to understand if confirmed hits are promiscuous?
Yeah, we have, so at Beacon we've run, I'm guessing it's more than a couple hundred HTS screens. So we do have tons of data on our compounds internally, and we use that data to identify whether or not compounds are promiscuous in the HTS, and so that helps us pick which compounds to follow up for dose responses.
Great, and yeah, to follow up on that question there, are there any phys chem properties data available for confirmed hits? Any measured data, for example, solubility, to provide any confidence in the quality of a hit?
Yeah, that's another great question. We don't have solubilities measured for all our compounds, but as soon as we get the compounds in, especially if we get good dose response data, they start to get evaluated for ADME PK properties right away.
Great, thanks, Carlton. So how quickly can one go from an idea to a screen and to potentially hits?
So that's a great question. It's a little bit tough to answer. So if an idea is that you have a receptor that you want to screen, if you have assays available, and they're robust enough to run an HTS on, then running an HTS takes, the screen itself is pretty rapid, as you saw in my presentation. That's 2 or 3 weeks to get the HTS done. Then of course it takes another 2 or 3 weeks to do the follow-up assays, and then when you have to acquire compounds, that takes a little bit longer. If you step a little bit further back, if you have to build the assays, and we have pretty strict requirements for our assays, they have to be robust, they have to have a good signal to noise parameters before we'll even start an HTS. So it can take maybe another month or two on the front end to have the assays. Now for a virtual screen, as long as you've got a crystal structure or a pretty good homology model, you can relatively rapidly, say in the space of 2 or 3 weeks, put together a model, do the virtual screen in a week or so, and then it takes 5 or 6 weeks to have the compounds in. And as long as you've got a good assay to test them against, you're ready to go.
And fantastic, thank you. So when are compound-focused virtual screens a better option than docking?
So that's another really good question. We've talked a little bit about having homology models and using those. Well, if the protein that you're trying to do a virtual screen on is far away from any template, and you try to build a homology model and the statistics around those homology model don't look like it's going to be an appropriate tool, if you have chemical matter for that receptor available, so compounds that you know or been shown in the literature to modulate that receptor, those ligands are a very powerful tool to enable a virtual screen.
Great, and next question now for you, Carlton. Is the high throughput screen library available as solid for a fast turnaround time from liquid XC50 confirmation to solid validation, or would these need to be synthesized? And then another question there, or are the majority commercially available?
Yeah, so that's another great question. So most of the compounds in the library are already dissolved and they're in DMSO. Probably 70 to 80% of them they're commercially available, and we've got a really good chemistry group that can synthesize the compounds that are hits that we aren't able to get to purchase.
Fantastic, thank you. Well, thank you very much, Carlton, for your excellent answers there. And unfortunately, that is all we have time for, but please join me in thanking Carlton for his time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please do take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we'd greatly appreciate it. So now, on behalf of Drug Target Review and Beacon Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.